Welcome to this biochemistry lecture on amino acid catabolism. So today we're going to focus on the right-hand side of this graphic where we talk about proteins and how they enter into cellular respiration. And you can see that they do that at pyruvate, acetyl-CoA, and citric acid cycle. We're also going to talk about deamination and the urea cycle, as well as heme catabolism, just because it's the logical place for it to occur. So proteins can be used either as building blocks, we can break down proteins into their constituent amino acids, and then they can be used to rebuild proteins, or we can break them down all the way and use them for energy. And about 75% are used to rebuild proteins, and about 25% are used for generating energy. It's a continuous process. We turn over 1 to 2% of our body protein every day. It's primarily muscle, and it does vary widely between individuals, but it's an important part of your entire metabolic processes. Digestive enzymes and plasma proteins are also rapidly degraded, and it also occurs in tissue that are undergoing rearrangement. So if you're, for example, if you're working out a lot, those proteins get rebuilt. If you're pregnant, some of your uterine tissue and skeletal muscle occurs. If you're starving, there's other things that have, skeletal muscle gets degraded. Some of the metabolic stable things like collagen can be measured, have half-life in terms of months or even years because we don't necessarily want to degrade those. Anyway, we will talk about how all this kind of plays into the biosynthesis of other compounds as well. There are two types of amino acids as far as the degradation. There are glucogenic amino acid. It's an amino acid that can go into glucose and subsequently can go into gluconeogenesis. And then there are ketogenic amino acids, which are converted into ketone bodies. So ketogenic amino acids are broken down into acetoacetate or acetyl-CoA. We've talked about the ketone bodies earlier when we talked about glycolysis. Some of the larger amino acids, tryptophan, phenylalanine, tyrosine, isoleucine, and threonine, are both glucogenic and ketogenic, meaning they can go in either pathway. And we have two that are purely ketogenic. They're lysine and leucine. And so all the others are glucogenic, and those can go, again, into glucose. So here we see how that occurs. We can have the amino acids in yellow can convert into acetyl-CoA or acetoacetyl-CoA, and those are the ketogenic, and they go into the citric acid cycle. After they're converted to acetyl-CoA by the ketone bodies, we also have the various amino acids that can go directly into the citric acid cycle at the spots you see located. So glutamine goes into as alpha-ketoglutarate, and there's enzymes that just convert all of these into the citric acid cycle. The amino group has to be removed, and that's how this occurs. So basically you get a deamination of the amino acid, and you have your carbon skeleton that allows you to enter the citric acid. That enters the citric acid cycle. So that happens by transamination, and transamination interconverts pairs of alpha amino acids and alpha keto acids, and during transamination the amino group of the amino acid is transferred to a keto acid, and which produces a new keto acid, and the original keto acid forms an amino acid. So this is the transamination reaction, and you get a transaminase that does that, and you can see that in this group, this graphic. One other coenzyme, peroxidyl phosphate PLP, is present at the catalytic site of aminotransferases, and that aminotransferase is the enzyme. PLP is a cofactor. It's a derivative of vitamin B6. And during transamination, PLP serves as the carrier for the amino group. And so you can see that's how you can see that in this group. PLP will convert alpha-ketoglutarate into L-glutamate and an L-amino acid into an alpha-keto acid. And so that's how all of these occur. PLP forms a shift base with a second keto acid. So that's the organic chemistry behind it. Transamination is used for the catabolic as well as anabolic processes. So we see this almost everywhere. So there are two mechanisms that are available for the transport of the ammonia group, the nitrogen group, from the peripheral cells to the liver. The first uses glutamine, and you can see the glutamine is the product that comes out of the muscle cells. So muscle has amino acids. There's a transdeamination reaction. The amine group is transferred to a glutamine by glutamine synthetase, and then it gets transferred to the liver. The second is primarily used by muscles and involves transamination of pyruvate to alanine, and then that alanine gets transferred. Once it gets to the liver, glutamine gets converted, gets deaminated into glutamate, and tyrosine can directly go in there through tyrosine aminotransferase, and that then heads into the urea cycle. So those are the mechanisms by which muscles and ammonia is transferred to the liver basically for removal. And here's a nice little spot of glutamate getting transferred. It's the glutamine or the glutaminase, and it's a ATP catalyzed enzymatic reaction. So amine groups are move freely by transamination. So PLP forms an amine, and the amino with the amino group of an amino acid, and then you get a rearrangement of the amine, and that gives an isomeric amine, which you see in the middle. And the hydrolysis of that isomeric amine gives a keto acid and peroxidamine, and then peroxidamine transfers that amine group to another alpha keto acid. So all of these reactions are to transfer that amine group around until it can get to its final destination, which usually is the urea cycle. Nitrogen atoms are excreted. Before that, they're collected in glutamate and glutamine, then is oxidized to alpha-ketoglutarate and the ammonia, and that conversion of glutamate to alpha-ketoglutarate is an example of an oxidative deamination reaction. And from there, that amine group enters into the urea cycle. So glutamate transfers with water. There's an NADH involved as a product, and we get alpha-ketoglutarate and an amine group as a product. And then that amine group, that ammonia, basically, I should say, the ammonia enters into the urea cycle. Arginine can also enter into the urea cycle, and this is kind of, you have your citric acid cycle that's going on in the mitochondrial matrix. From there, you can do things can either enter or exit the citric acid cycle. Amino acids can through transamination, so you can have alpha-ketoglutarate and aspartate that are produced from glutamate. That aspartate can enter into the urea cycle. That aspartate gets transferred into arginosuccinate, which transferred into arginine, which produces urea and ornithine, and from there the ornithine can get recycled and go back into the urea acid cycle. And so the urea cycle is also known as the ornithine cycle, and it occurs in most animals, and the products are urea, and the products are urea, and the reactants are ammonia. Generally, ammonia is not easy to get rid of, and so that's why we have urea, and we'll talk about that here in a little bit. It occurs in stages, so you get a transdeamination, which is a removal of the alpha amino group through transamination and deamination, which basically means we transfer or we get rid of. Transamination means we're going to take it from one molecule to another. Deamination means that we have ammonia as the product. Glutamate in peripheral cells gets deaminated and forms ammonia in the liver. And we have another minor pathway where we have oxidative and non-oxidative deamination in peripheral cells to produce ammonia. And then we have a third option where ammonia is transported to the liver as glutamate, glutamine, or alanine. And then we get detoxification of that in the urea cycle, and ammonia is converted into urea. So if you have insufficiency of the urea cycle when in some metabolic orders, and that is a really serious issue where a lot of development issues occur. So urea is significantly less toxic than ammonia, and the urea cycle, which occurs mainly in the liver, is a less toxic way of getting rid of nitrogen. The nitrogen atom, you can see the structure here. Ammonia is a straight amine group, also NH4, which is an NH4 plus, or we can put it onto a carbon backbone, as you see here, with a ketone, with a bond there on that carbon, and that can get transferred, and that's how it gets transported out of the body. So the urea cycle is a cyclic pathway, produces urea from carbon dioxide and ammonia, and in that it's going to use energy. So we start with we start with carbon and ammonia. We use 2 ATP. We generate 2 ATP or ADP in water. That goes to carbamoyl phosphate. Carbamoyl phosphate enters in with aspartate, when we get a transamination reaction where ammonia is produced, and we end up with fumarate. Fumarate then gets transferred to urea. Here's how arginine enters the process and is involved in the process. So we basically convert urea in the liver, and that from there we go arginine to ornithine, and we produce a urea. Ornithine gets transferred to citrulline with carbamoyl phosphate. Citrulline with aspartate goes to arginosuccinate, and then that goes into fumarate, and from there we recycle the whole process all over again. Again, you can see here's some specific reactions where we're going from ornithine to citrulline, where we're transferring that phosphate group that gives it the power. That power then allows for the transfer, the transaminase reaction to occur, where we take aspartate, it gets transaminase to arginosuccinate. Arginosuccinate then goes with fumarate, where we do another transamination reaction to go to arginine. Arginine then gets deaminated to urea, and the products are urea and ornithine. That's the reaction. So we're going to very briefly mention heme catabolism. That's the end of the urea cycle. I would know that for your exam. So we'll just basically cover heme catabolism right now because heme is associated with the large proteins. And you will see this on your exam. So heme gets degraded from heme to biliverdin to bilirubin. And all of them have different color profiles. Heme is red or blue depending on the oxygenation state. Biliverdin is very green as it degrades, and then bilirubin is very brown. And so as you get a bruise, you're going to have different coloration based on the amount of these molecules that you have. So globin is hydrolyzed to amino acids and then reused. Iron is preserved in ferritin. When we start having a degradation, ferritin is an iron-carrying protocol. Heme is then converted into bilirubin. Bilirubin enters the liver and then is transferred to the gallbladder, where it's stored as bile, and then it's finally excreted into the feces, where it's very brown, and that's why you have brown feces. And with that, we are done with this lecture. Thanks for listening. Look forward to hearing you on the next slide.